In this video, I'd like to discuss proteins a little bit further. We're going to go into depth on protein structure. And it's going to turn out that protein structure is really important for the function of proteins. So the first thing we need to discuss is the fact that proteins do a lot of things for you. As I mentioned in a previous video, some proteins are fibrous and they provide strength. So for example, you have proteins in your skin, keratin's an example. And it provides a lot of strength to your skin. Keratin happens to be found in nails and hair also, but that's just one example. So proteins give structural strength. Some proteins, as I mentioned previously in a video, are like little machines. So some proteins are going to move substances across cell membranes, and that's of course very important for your cells to function properly. Other proteins will speed up chemical reactions. They'll act as what's called a catalyst. These proteins are called enzymes, and we're going to go into that in further detail in another video. The structure of a protein has different levels of organization. The first level of organization is called the primary structure. The primary structure is simply the sequence of amino acids. Remember that proteins are polypeptides. Polypeptides are chains of amino acids. Each individual amino acid is covalently bonded to another one. And we talked about this. The process of building this chain is through dehydration synthesis. Each amino acid has a specific three-letter code. So for example, we can see here in the diagram next to me with the primary structure, which again is the sequence of amino acids. And we have a number of amino acids here, such as arginine is the first one. Okay, let's go on and move on to the next slide. So the next level of organization is the secondary structure. The secondary structure is the three-dimensional shape of local segments of protein. So as the protein is being made by adding successive amino acids to the chain may spontaneously form a three-dimensional shape. And so certain sequences of amino acids will lead to the formation of what's called an alpha helix. The term helix means twisted or curved. So this is a very common sort of structure that's found within some proteins, these little segments in which you have a helical structure, this alpha helix. If we look at the alpha helix, well, what's causing the alpha helix? Well, one of the major forces that causes the alpha helix is a pattern of hydrogen bonds between the hydrogen and carboxyl, the amino hydrogen and carboxyl oxygen atoms within the peptide backbone. So we can see that here in this figure here. You can see hydrogen, which is attached to nitrogen. So that's the amino hydrogen. And it's making a hydrogen bond, which is showed with a dashed line, to an oxygen, which is formerly part of a carboxyl group, right? Because you can see the oxygen is bound to carbon. Now that's one very common form of secondary structure, which is this alpha helix. It's found, of course, in human proteins. It's found in plant proteins. It's found in bacterial proteins. Very common structure. Another very common secondary structure is called the beta pleated sheet. Sometimes it's simply referred to as the beta sheet. And you can see an example of that right above me. And so this is a little bit different. It forms sort of a sheet-like structure. And in which there's two possibilities. One is the chain may bend back on itself, or it actually could be separate chains that are actually close to each other, and they're going to form this beta pleated sheet. And again, you can see hydrogen bonding between the amino hydrogen on one amino acid subunit to a oxygen from what was formerly a carboxyl group on another amino acid. Okay, a third level of structure is called the tertiary structure. It's the overall three-dimensional shape of a protein. So this is dependent on interactions between amino acids within the chain. It's also dependent on interactions between amino acids and the aqueous environment. So here's an example. Perhaps you have some amino acids in the chain with R groups that are polar or charged. Because they're polar or charged, they'll be hydrophilic, meaning that they're water-loving. They'll tend to interact with water molecules, which are polar. And so these amino acids tend to be located on the outside surface of the protein and sort of pointed outwards. Now, you may have some other amino acids that have nonpolar R groups. Because they're nonpolar, they're going to be hydrophobic, meaning they don't really like to associate with water. They do, however, like to interact with other amino acids that are also have nonpolar R groups. So these amino acids will tend to be on the interior of a protein, sort of pointed inwards. So these are and there's, of course, these are just some, but there are all other types of interactions. But these are certainly very important. These hydrophilic and hydrophobic interactions are very important for the three-dimensional structure of a protein, which is again called the tertiary structure. And you can see actually in this picture beside me that we can see the primary structure, which is the actual linear sequence, right? It's the sequence of amino acids. You have the secondary structure, which is things like local areas in which you have an alpha helix or a beta pleated sheet. And then the tertiary structure is the overall three-dimensional structure, which is much more complicated. Now, that's it for many proteins. A lot will just stop. The tertiary structure is the last level of organization. But there are some proteins that actually have a fourth level of organization. This is called the quaternary structure. A quaternary structure of a protein occurs when a protein consists of more than one polypeptide chain. And there are many examples, but the typical one you find in textbooks is that of hemoglobin. Hemoglobin is a molecule, it's a protein that has a quaternary structure. It actually has 4 subunits that are interacting to make one hemoglobin molecule. Hemoglobin is found in red blood cells and helps to transport oxygen. It consists of 4 subunits. Each subunit is a polypeptide. All right, so let's move on to the next thing. So I've talked about primary structure. That's the linear sequence of amino acids. I've talked about secondary structure. That's as the protein is forming, it'll actually start to make things like an alpha helix, or it could make a beta pleated sheet. Potentially, you could actually have a beta pleated sheet between separate polypeptides also. And then finally, we have the third level, the tertiary level, in which you have hydrophilic and hydrophobic interactions that are causing a protein to have a specific three-dimensional structure. And then on top of that, if you have a protein which actually consists of multiple polypeptides, that type of protein would have a quaternary structure. Now, why is this so important? Well, protein function depends on structure. And one thing I want to clear up right now, because we're going to talk about this in more detail in other videos, but one misconception is that proteins always have this sort of rigid, unchanging three-dimensional structure. That's not true at all. Proteins can change shape in the presence of polar or charged molecules. So, and why do they have this ability to sort of be somewhat flexible? Well, it's because a lot of the shape of the three-dimensional shape of a protein is actually dependent on relatively weak bonds, such as hydrogen bonds, interactions, as I said before, between hydrophilic side chains and water, and hydrophobic interactions within the protein itself. So none of those are really as strong as the primary structure, which is the covalent bonds. But because of that, the proteins are actually able to change shape a bit. And that's really important for their ability to actually function. And here's an example that I made up here. We have a protein, and it has a site where it can accept another molecule. But perhaps that molecule doesn't quite fit exactly into that site. But once the molecule, which might have some charged groups on it, interacts with the protein can actually change shape and sort of, move to fit the molecule better. And I'm using this example because it's going to be important when we talk about enzymes. So again, I just want to throw this out there that proteins are not static. They can actually move, and that's very important for their ability to do their functions within your cells. Okay, that's it for this video. Thank you for watching.